This evening, you have given me 3 minutes of your time, 180 seconds. Every 98 seconds, an American becomes the victim of a sexual assault. Look around you. 1 out of every 6 women have been sexually assaulted, and 99.4% of their perpetrators walk free. You see, when a sexual assault occurs, we have 3 days to gather the biological evidence before it degrades. The collected swabs are assembled into a kit that are sent to a crime lab where they will wait 3 months, along with tens of thousands to 500,000 other kits in the US rape kit backlog. That's 3 months of time in which the perpetrator is able to walk free, committing the same crime to countless other victims, as estimates of up to 90% of rapists are repeat offenders. Why does it take us this long? Well, the primary challenge in rape kit analysis is the separation of male from female DNA from the collected swab. Since the swab was taken from the body of the victim, if analyzed directly, it will yield an overwhelmingly female profile. Therefore, the initial separation of male cells from the swab prior to analysis is essential for perpetrator identification. But the current methods are time-consuming and labor-intensive. They consist of a series of overnight incubation procedures and sample transfer steps that introduce the risk for sample contamination or sample loss. At best, these methods provide us a 30% chance of successfully identifying the perpetrator. My work is the invention of a novel technology. I use a technique called capillary zone electrophoresis to perform this separation. I use a silica capillary. It's very similar to the size and shape of a strand of your hair. I inject the sample into one end, apply a voltage onto either end, and I'm able to separate out different cell types by their unique size and charge ratio. I've coupled this system with an automated fraction collector that I've programmed to deposit purified aliquots of your male cells, female cells, and free DNA from the swab. Compared to your week-long method in a crime lab, my separation takes 15 minutes, with the objective of identifying the perpetrator the afternoon of a crime and stopping him from committing future assaults. I've reached out to and spoken at crime labs from New York City to LAPD to Honolulu Police Department in order to optimize and integrate my technology into their workflow. But it's going to take more than research scientists and crime lab analysts to end the rape kits backlog. Indeed, a challenge of this magnitude demands policy and legislative changes that can only be effected with the support of our entire community. Therefore, this evening, I invite each one of you to get involved. Would you reach out to learn more, take a stand, and join me in battling the backlog?